Applications of photocatalytic oxidation coatings. Learning objectives. To understand benefits of using photocatalyst oxidation coatings on surfaces. To understand the chemistry of photocatalyst oxidation coatings. To illustrate applications and provide sufficient information for a designer to make a recommendation to a client. Environment and health issues. Some health concerns of today's society include maintaining healthy environments, pollutants from industry and vehicles, poor indoor air quality, contracting infections from health service areas, and creating an environment-friendly workplace. Due to increasing human population, more areas are becoming increasingly urbanized, which also increases pollutants caused by industry and fossil fuel burning vehicles. Increasing population also need housing, which require materials manufactured and constructed with products that emit harmful vapors and fumes. Along with the increasing human population, viruses and bacteria also can spread more rapidly and widespread than in the past. Hospitals and assisted living facilities continue to struggle to control nosocomial infections, which cost the healthcare community in the U.S. alone millions of dollars annually. And cleaning and maintaining affected surfaces can be costly for businesses and individuals. Maintenance costs increase as time passes. The solution for these concerns is photocatalytic oxidizing coatings, which, along with light and water, have the ability to keep surfaces clean and microbe-free. What is photocatalytic oxidation? Photocatalytic oxidation can be described as the event of using light as a catalyst to create superoxides and hydroxyl radicals from oxygen and water. Semiconductor metals such as titanium dioxide use light and water as catalysts to activate movement of electrons, freeing them for secondary reactions with harmful organic molecules such as those found in VOCs, fossil fuel emissions, and bioaerosols. How small is a nano? A nanometer is 1/1000th of a meter. Photocatalytic oxidizer nanoparticles are about 10 nanometers in length. How does that size compare to other microscopic items? Consider the size of a pinhead, which has a diameter of 1 to 2 millimeters. A dust mite has a length of around 200 micrometers. Red blood cells have a diameter of 7 to 8 micrometers. And DNA strands have a diameter of just over 2 nanometers. Reviewing this concept graphically, we find a molecule of a titanium dioxide nanoparticle which has a band gap energy of 3.2 electron volts between its valence band and conduction band. When the titanium dioxide nanoparticle is irradiated with light photons of less than 385 nanometers, the band gap energy is exceeded and an electron is promoted from the valence band to the conduction band. The resultant electron-hole pair has a lifetime in the space charge region that enables its participation in chemical reactions. These chemical reactions include the formation of superoxide ions caused by the combination of the free electron and oxygen found in the air, and the formation of hydroxyl radicals caused by the loss of a hydrogen electron from water found in the air. Hydroxyl radicals and superoxide ions are highly reactive species that will oxidize volatile organic compounds and organic pollutants absorbed on the catalyst surface into primarily carbon dioxide, water, and inert minerals. They will also kill and decompose absorbed bioaerosols, making the process a strong candidate for indoor air quality applications. Additionally, hydroxyl radicals have a high oxidation potential, meaning it takes less of an effort to donate an electron to organic molecules. The oxidation potential of hydroxyl radicals is much greater than the common disinfectant chlorine. Properties of photocatalytic oxidizers. When photocatalytic oxidizers are activated with light, treated surfaces display properties that are both environment-friendly and cost-beneficial. Surfaces become anti-organic. Treated surfaces removes volatile organic compounds disengaging bioaerosols. Self-cleaning. Treated surfaces create a superhydrophilic surface that allows water to separate dirt from the surface, minimizing cleaning time. Deodorizing. Because odors are caused by bacteria and other organics, treated surfaces provide a continuous, natural deodorizing property. Anti-organic properties. Comparison of common disinfection methods to photocatalytic oxidation. To demonstrate the effectiveness of PCOs as an anti-organic treatment, we can compare PCOs to common disinfection methods many of us use to control organics such as viruses, bacteria, mold, mildews, and algae. Common disinfection methods include the use of chlorine, which is toxic not only to organics, but also to animals and plants. Although disinfectants are effective in deactivating organics, the results are not permanent. Microbes that are not deactivated will again multiply when the disinfectants are no longer present. Most disinfectants are also, by their very nature, potentially harmful and even toxic to humans or animals. Because disinfectants are designed to kill, have a great potential to cause DNA damage to animal cells, cell mutations, and ultimately cancer. The PCO methods greatly reduce harmful effects to the environment and to humans and animals. PCOs do not use toxic chemicals. Products found in PCOs such as titanium dioxide are also used in common everyday products such as cosmetics. PCOs provide a continual deactivation of organics on their surfaces. All microbes are deactivated so they cannot reestablish colonies. Because the byproducts of PCOs are primarily carbon dioxide, water, and inert minerals, they are not toxic to humans or animals. Unlike other disinfection methods, PCO high redox potential doesn't interfere with human or animal cells. Because they do not produce vapors or fumes like some common disinfectants, PCOs create a healthier, safer environment. Decomposition of bacteria cells using photocatalyst oxidizers. Lab tests indicate the effects that PCO-coated surfaces have on bacteria. E. coli and MRSA bacteria were applied to PCO-coated ceramic tiles as well as uncoated ceramic tiles and then placed under a light source for 24 hours. Results of the test show a high cell count remaining for both types of bacteria on the uncoated surfaces. In contrast, the coated surfaces show bacteria cell counts were reduced to virtually nil. Bacterial decomposition on PCO-coated tiles. Additional bacteria and viruses were tested on PCO-coated tiles with the same positive results. Photos on the left of each picture show the results of coated tiles after 25 minutes of light exposure. Photos on the right of each picture shows the results of uncoated tiles after the same time period. Decomposition of a bacteria cell. How do PCOs deactivate bacteria and virus cells? PCOs deactivate cells by attacking the cell walls, which are comprised of sugar molecules, and converting those molecules into carbon dioxide and water. Once the cell wall is compromised, the interior of the cell, the cytoplasm, leaks from the cell, rendering it deactive. Real-world results in a surgery room. Tests have been performed in real surgery rooms treated with photocatalytic oxidizers on floors and walls. Samples of bacteria on walls and floor, as well as airborne samples, were taken in a surgery room prior to application of photocatalytic oxidizers. Both bacteria counts were at a high rate prior to application, but dropped drastically after the installation of PCOs to the walls and floor. Real-world results influenza case study. During the influenza seasons between the years 2003 and 2006, tests were conducted in 2 wards of Nagano Prefecture Kiso Medical Facility in Japan on a 50-bed PCO-treated facility and 50-bed untreated facility. Between the months of October and March of each test year, the percentage of patients who contracted influenza was recorded for each ward. The results show that nearly 9% of the patients in the untreated ward contracted influenza, but only 1% of the patients contracted influenza in the treated ward.